Okay, welcome back to Biological Anthropology. This lesson is on the basics of genetics. So, you've probably previously learned about the history of evolutionary thought, especially about Darwin and the fact that Darwin discovered natural selection. Well, Darwin and Wallace discovered natural selection, but one thing they didn't know about is exactly how traits were inherited across generations. And now we know that they are inherited through genes, but the key point is that Darwin didn't know about genetics. And this is what we're going to do with this lesson, is kind of begin to bridge concepts of natural selection with concepts of genetics, and in the next few weeks, build and realize today's modern evolutionary theory. So hopefully you remember from earlier in the semester that biological evolution is a change in gene frequencies in a population over generations or over time. So we're going to learn more about the genetic aspect of that definition in this lesson. First, by reviewing some basics of genetics. Okay, it was briefly mentioned, probably in your textbook, that Gregor Mendel discovered basic principles of heredity. He had deduced the presence and activity of genes, which are portions of DNA that direct the synthesis of specific proteins. So let's clarify here the difference between genes and DNA. Well, there isn't really a big difference, but a gene is basically a segment of DNA that does something. Here you have a more specific definition that says a portion of DNA that directs the synthesis of specific proteins. So there are different genes that do different things. Some genes make proteins, some genes regulate, but a gene is basically a functional portion of DNA. Since everything in the body is made up of or by proteins, genes code for every part of an organism. So the main job of genes is to synthesize proteins or have to do with processes involving proteins. Okay. There can be different forms of a gene. These different forms of a single gene are called alleles. For example, the gene for a human blood type can exist as an A allele or a B allele. So the genes, the different alleles are doing the same type of job in a different way, a different variety, okay? Remember that variety is required for natural selection to occur. Hopefully that sounds familiar. The human genome, which is the complete sequence of human DNA, contains 3 billion chemical bases. So those are the letters A, G, T, or C, and 20,000 to 25,000 genes. Don't forget, we learned what genes were a few slides ago. So it takes billions of letters to come up with the genes that regulate everything in our bodies and build all these proteins and put us together. A gene frequency, remember, the definition of biological evolution is a change in gene frequencies in a population over time. A gene frequency is a proportion of genes or alleles of different types in a population. For example, a very simple example could be the proportion of brown-eyed alleles to blue-eyed alleles. That would be a gene frequency. Even though, in reality, many different genes will code for eye color, which makes it a polygenic trait. But our simple example is a proportion of brown-eyed to blue-eyed. That would be a gene frequency. Okay, let's review Mendel and what he discovered about genetics. So, Mendelian inheritance views heredity as based on non-blending single-particle genetic inheritance. This is based on the work of Austrian monk Gregor Mendel, who lived from 1822 to 1884. He bred pea plants and discovered how genetics or inheritance work. He showed that traits can be passed on through the generations. Dominant traits can be expressed in an organism. Recessive traits can often not be expressed in an organism. So the way that different traits are expressed can vary. He also discovered the principle of segregation, which means that individuals receive one gene for each trait from a parent, and that each gene is half of the required pair for each trait. He also discovered the principle of independent assortment, which means that each pair of genes separates independently of every other pair when egg and sperm are formed. So these questions or these facts might be in your quiz. This is just a review of what Mendel discovered. So how did he do this? He crossbred peas with red flowers and peas with white flowers. This produced the next generation. And he discovered the dominance and recessive factors or traits in these pea plants by breeding and crossbreeding over and over with his experiments. Okay, more on modern genetics. Let's review a little bit more, and the next segment of our lesson is going to be about DNA replication. Okay, so how does DNA provide instructions for creating proteins? DNA is converted to RNA through a process called transcription. And then the RNA is used as the instructions to create amino acids which string together and connect to build proteins in a process called translation. So transcription is when DNA is converted to RNA, and translation is when RNA is used to build proteins. Here are some other important terms that you should know. Again, in the basics of genetics. Hopefully a lot of this is review if you've ever taken biology. So homozygotes means a fertilized egg that receives the same allele for a specific trait from each parent. So homozygous just means an organism carries 2 copies of the same allele. Okay, it doesn't necessarily have to be in an egg. In this situation, kind of ignore that part. Heterozygous means that there are different varieties, or different alleles for a specific trait. Mitosis is the way body cells replicate or copy themselves. Meiosis is the way sex cells replicate themselves. And we're going to talk more about that in the next segment of this lesson.